Well, welcome everyone to the fall event sponsored by Upstate New York Society for Risk Analysis. I'm Peg Coleman, the president of the regional organization. We have virtual audiences today through live streaming on Facebook and Zoom. The program is packed, so I'm going to make short introductions of both speakers. After they speak, I'll return to the podium to frame the discussion, and please hold your questions until then. By 6 o'clock, we'll break for refreshments just outside the door and informal discussions. Our first speaker is Rodney Dietert, professor of immunology at Cornell University. He earned his degrees at Southern Methodist University, Duke, and University of Texas, Austin. Rod is also a prolific author, and I had the pleasure of getting his signature on this 2016 book of his. The book will be on display. There's a display table outside the room, and those materials are there for browsing while we have our informal discussion. Rod, I just learned today that you are an official microbiome hero. This honor celebrates the first World Microbiome Day, reminding us to mind our microbes. Thank you for taking the time to prepare what I know will be a stimulating talk for us today, Rod. Now, our second speaker is Catherine McComas, professor of communication and faculty fellow of the Atkinson Center for a Sustainable Future here at Cornell. Catherine also serves as area editor for the SRA journal Risk Analysis, and there are some of these journal volumes are displayed outside. Catherine is area editor for the SRA journal in the risk communication area. She also has the mixed blessing of serving as president-elect of the International Society for Risk Analysis, taking over the gavel to serve for a year as SRA president at the December meeting in New Orleans. Her LinkedIn profile lists that Catherine cares about education, environment, health, science, and technology. One of her papers of special interest to this audience is this one. I'll also put this for display outside. The title is Public Understanding of One Health Messages, the Role of Temporal Framing. Thank you, Catherine, for serving as rapporteur today and for most excellent work in risk communication. Now, if Rodney would like to come up to the podium.
Well, thank you, Peg. It's an honor to be able to participate in this symposium workshop, to join Catherine on the program. And I really want to thank the Society for Risk Analysis for this opportunity. It's really important membership, work, education, and progress in protecting humans, animals, plants, and Earth in terms of all of the risks involved and understanding risk benefits. So it's an extremely important area. And Cornell is blessed to have faculty that contribute to it across a number of our colleges. So thank you for this opportunity to join in. Disclosures: in addition to being a professor here in the College of Veterinary Medicine, and I've just entered my 42nd year as a faculty member at Cornell, I am recently a consultant in education translational research with the new probiotics company Seed, and also an author with Dutton Penguin Random House. This is the first time I've ever done this, and that is to make a prediction. And the prediction is that in the near future, the highest priority for both preventive and therapeutic medicine will be managing microbes. And I hope that by the time I'm toward the end of the talk, I'll have some justification for making that prediction. Because I think we really are going to get to a point where we're going to be master gardeners of our microbiota. We're going to be the shepherds of the majority genes in us, which will be from the microbiome. So I'll try to explain why I believe this is really where we're headed, and as soon as we can embrace that in terms of benefit risk, the better. So I've got this divided sort of into 4 parts. The first is an introduction, an explanation about the human superorganism, and that is the mammalian part of us plus the microbial part. The microbiome as our gatekeeper, and I'll try to show you how toxicology, infectious diseases, and the microbiome all collide in this idea. Talk about the microbiome and the immune system and how it operates in health and in disease, and what we can do to try to keep it in health. And then finally, breastfeeding and human breast milk is a way to introduce some of the discussions that we'll have subsequent to the presentations. So, by saying that humans are superorganisms and that our microbiome, which are our bacteria, viruses, archaea, protists, fungi, that they are a huge part of us in health and disease, we're not really focused just on humans. In fact, Earth itself and the upper atmosphere of Earth and the International Space Station is cultivating microbes and has we're a microbial planet. It has a bubble around us. We carry a bubble with us to hotel rooms and to classrooms and to our offices. As we go, we leave some, we probably pick up some. Our animals help transport them between us as well. So this is the reality. We've only come to really appreciate it, although there are bacteriologists from back in the 60s, even some here at Cornell, that could have told us we should have been focused here in the first place. So I got into the microbiome by accident. I was working in the developing immune system and how to protect that for decades here at Cornell. And I got a challenge I couldn't resist, and that was to identify in newborns, in babies, the single most useful or biomarker, we would say, from toxicology, or measure, or sign, that you could measure in a new baby that would best predict whether that baby had a life filled with health or one filled with disease. Now, a lot of things happen across a lifetime. You eat different things, you get exposed to different drugs, environmental chemicals, and the like. Those all have effects. But the question is, what could you measure then that would be the best predictor? And I was pretty sure I knew, only when I started to write the paper, I realized I didn't know. Went to bed really frustrated that night, woke up in the middle of the night with a very simple idea. Woke up from a dream, and the idea was that the baby has to self-complete. And self-completing with the microbiome, which is just a slight majority of cells and a vast majority of genes for that baby, and the extent to which that baby self-completes would be a really useful measure, would probably be the best predictor at that stage. And so flushing this out over subsequent papers and the like really threw me into the world of the microbiome, even though I'd not been there based strictly on my research in prior decades. So that is that the newborn really needs to acquire what it turns out are family-sourced, largely, microbes, and those get installed, and I'll talk about where our microbiome is located in the body. It's very interesting to me as a toxicologist. And self-completion, and this includes major events, such as, well, first of all, the prenatal period, where the placenta is now known to have a microbiome, and it has influences on the developing, on the fetus. But natural childbirth, vaginal delivery, skin-skin contact, all of those with natural childbirth come into big play with seeding the baby. And then breastfeeding. Breastfeeding is extremely important in several ways relative to what happens to the infant microbiome and what happens for risk of disease, as we'll describe. So that is the completed self for the baby. And it turns out that what is actually happening is that the human genome, you may remember the Human Genome Project, which showed a lot of promise, and has some, but showed a lot of promise for curing human disease. And it turns out that the reality of the numbers of genes versus the estimates were really a little bit less than half. So there are 22 to 25,000 chromosomal genes in humans. We're just barely a couple thousand more than the earthworm. So we shouldn't get too arrogant about things, really. We should be humble. Whereas with the microbiome, across all humans, they're just under 10 million. And in a given individual, we're carrying around about 3.3 million genes from our microbes. Now these genes are doing things just like our chromosomal genes are doing things. They're making enzymes, they're making a variety of signal factors. There's a huge amount of metabolism that goes on out of what is more than 99% of our genes, which are microbial. Based on cell number, people used to think it was 90% microbial. It's more like just over 50%, probably around 57% microbial cells versus our mammalian human cells. So by several measures, we're either a little bit majority microbial or a lot. And the genes count. And these genes are great because they're malleable. You can do something about it. Chromosomal genes, a little bit harder to change those out. Not that you can't, but hard. So one of the reasons to focus on things like what happens at birth and breastfeeding, early infant feeding, is because of the work of people like Dr. Barker, David Barker in the UK, and a group that I associated with for years when I was looking at the developing immune system. And that is what's called, emerged to be a society and a journal and everything, called The Developmental Origins of Adult Health and Disease. And NIH, and particularly National Institutes of Environmental Health Sciences, has been very active in this area, because what we know now is that prenatal development and early infancy and early childhood are critical windows when all of our systems are maturing. And depending on the system, some of them take longer. If you looked at lung, it takes a while before it's fully matured to adult form. But the immune system, the brain, neurological system, the gut, are all maturing. And there are events, benchmark events that have to happen, and they have to happen appropriately and at the right time and in the right sequence, or our physiology is off. It's just not right. And it's a little tricky to go back and correct it. So Barker realized that cardiovascular disease, that a lot of what happened was prenatal, early neonatal. And he started this idea of that developmental intervention, or the Barker hypothesis, is you really need to pay attention to what's going on during pregnancy and early infancy. And it turned out it's not just cardiovascular disease. You take any so-called chronic disease or noncommunicable disease and condition, and this holds true. So, as NIEHS has said in this figure, intervention in early life is worth a whole lot more than intervention in the adult. You're just simply more likely to be able to protect effectively and have it carry through than later on. It's not that you shouldn't do something when you're my age. I've done things that have been useful in a lot of cases, but you have to work a lot harder and change a lot more. And be lucky, maybe. Okay, so this shows, I'm not going to go into the details, but this shows here's the immune system and immune development, and you have important things happening. And we now realize when I was growing up here as a faculty member in the late 70s and early 80s, everything was the bone marrow and the thymus, and in chickens, the bursa fabricius, and what we call the primary lymphoid organs. Now we realize the site of action is where the immune system and the microbiome hit the road, at our barriers where they're communicating. So there you have specialized T lymphocyte maturation. You have memory cell generation occurring, again, largely in the periphery. And the interactions in our tissues and at these barrier sites is really important. And so a lot of this actually is happening prenatal, or I mean postnatal, early postnatal development, which means there are things that we can do to protect if we understand the relationships there. So in terms of the microbiome, again, the bacteria, archaea, viruses, protists, fungi, these effects, there are some prenatal effects. There's some debate whether the fetus is seeded at all. It may or may not be. But certainly the placenta and the maternal microbiome has influences on the developing fetus. Then the major event, seeding at birth, again, ideally vaginal delivery when that's possible, skin-skin contact to transfer microbes. And the option are, if that doesn't happen, where does the baby get the microbes? From the hospital or the general environmental area. It may not be what you want, seeding your baby. And then finally, breastfeeding. And breastfeeding has not just additional microbes, but also food for the microbes. The nutritionists and people that looked at it said, well, so much of breast milk you can't even use. Mammalian cells can't use it. They can't digest it. And that's because it's not there for our mammalian cells. It never was. It's there exclusively for the microbes. That should tell us something about how important the microbes are. That millennia of selection of breast milk leaves a lot of the content exclusively for the microbes. Okay, so we in Western countries have lost about estimated 35 to 40% of our diversity of the microbiome. And this has occurred largely through a whole variety of decisions, some that look brilliant at the time and not so brilliant now. Unintentional consequences of a lot of things. I'll show you a few. But this is in comparison to indigenous people in the Amazon region. So they don't have a lot of the non-communicable diseases that we have. And they have a much richer microbiome. But they can get to where we are. They can have cancer, obesity, diabetes, and heart disease by moving, within a couple generations, by moving to the suburbs of Sao Paulo and then into downtown Sao Paulo and eating like we eat. And there you are. So it can be degraded, and it's a matter of understanding what things degrade it and trying to protect and trying to seed and feed at an early enough age to be meaningful across the lives. Okay, so here's some of the things that contributed to this. Antibiotic overuse, and that would be things like ear infections that may be viral, and if we waited a few more days, we'd know that, but you show up at the doctor, you leave with a prescription, is really sort of an expectation. In addition, something that I railed against back in the 90s, and that is agricultural use, prophylactic use as a growth promoter in poultry and swine, which was never a good idea. It has led to antibiotic resistance in the environment and zoonotically for diseases, infectious agents that infect us as well. Urbanization, the urban environment is a microbial wasteland. I think you'll see papers actually titled that. And that's just by assessment, it really is. So we think we're green, we're not so green, microbially, and that's a real problem. Because that's where most of the people are actually in some cases. Birth delivery mode, again, particularly cesarean is medically necessary in many cases, but where it is elective, you then are not seeding the baby as you would with vaginal delivery. So there are things that can be done once we know it and once we commit to it. Misdirected efforts of human safety. I've been involved with safety testing for the immune system most of my career. And part of the problem is it was always modeling, usually using rodents or other lab animals, or in vitro modeling for the human mammal, never including the microbes. And so you should realize that any drug, chemical, food additive that has, even current ones, and I'll show you one example, that has not been vetted from microbial safety should not be assumed to be safe. And in fact, internal company surveys of this in the pharmaceutical industry show that concern is warranted, that they do alter the microbiome, because they were never screened for it. And mammalian-only human medicine, educating physicians, health practitioners, to look first to the microbiome, because it's the majority, and because it is really the gateway to our environment. Okay, so what happens if you're depleted? So with electric cesarean C-section, there's altered immune function, altered cytokine production, and again, people have shown that there is seeding. The baby will fill up with some microbes. They won't be very diverse, and they're more likely to be things found in a hospital or whatever the setting was, or whoever was attending to the baby. With the growth promotion use in, again, antibiotics in agriculture that's occurred, I mentioned that it selects for antibiotic-resistant genes that can then be horizontally transferred as well, and can increase the risk for humans and the environment through zoonosis. In addition, animals in captivity, and actually this would include our lab animals or rodents over in our animal facility. They usually eat a restricted diet. Again, they're not wild and free. They're not getting exposed to a lot of things. There's sort of a control in terms of pathogens and microbes. And as a result, they have pretty low diversity of the microbiome. And guess where a lot of their microbes come from? They come from the animal handlers. So your lab technician is helping to populate the mice that are right over here. And in the zoo, the animal handler is populating the howler monkey that has, in Vietnam, 57 species of plants that it eats, and 1 in American zoos. So the microbiome is really amazing to me because of partly of where it's located. Now there are parts of us that are exposed to the external environment. And as a toxicologist, we would talk about that exposure to the external environment as routes of exposure to potentially hazardous environmental factors. This would be through contact, dermal exposure, through ingestion. You know, you're drinking arsenic-contaminated drinking water or lead-contaminated water in Flint, Michigan, right? Or inhalation. Particles, diesel exhaust particles, other things in urban environments, for example. And that's toxicological routes of exposure. Well, it's also, if you include urogenital, major portals of entry for infectious agents. Guess what? They come into the same place. It is also the major sites for the microbiome. So they are the first thing that sees these environmental factors. They basically are our gateway, our filter, and we see the world, our body sees the world through our microbiome in terms of what's outside of us. In addition to some things like breast tissue actually having its own microbiome as well. But nevertheless, there's skin microbes, there are microbes in the airways, nose, lungs, there are microbes all the way through the GI system. They're very specialized depending on the region. And urogenital as well. And these are, as it turns out, protecting us if we pay attention to risk-benefit. Otherwise, they're not. So your exposure, you have exposure to arsenic in a drinking water. Your actual internal exposure depends on your microbiome in the gut. And that single exposure can result in some very toxic metabolites or not, depending on your microbes. It's not to say arsenic's safe, it's just to say your individual risk depends upon your microbiome. And that'll be the same for drugs and for food additives and everything else. So what happens when microbes encounter a drug or a chemical or these factors, they do a lot of different things. They may hoard it, they sequester it. They will avoid it, they don't want to see it. Exclude it, they'll metabolize it and make a variety of metabolites. They will signal to other microbes or to our epithelial layer. They will die in some cases. They'll kill some microbes. Or they'll grow. They can use it, and they will. Or worse, they'll translocate, because microbes moving from one ecological niche in our body to another usually leads to pathology. It's not a good idea in general. So we have all of these responses available. And in addition, I mentioned that they have tremendous metabolic potential, plus they can epigenetically regulate our liver on its metabolic potential. And you can see people who spent their whole career, like Curtis Clausen, a prior president of Society of Toxicology, a terrific liver metabolism person, who's now writing about the microbiome metabolism because that's where tremendous opportunities exist, and again, the metabolic potential is as vast as the liver. And that gives us a chance to do something useful in terms of human metabolism and food. So, gut microbes can increase or decrease drug availability. They can metabolize the drug, and a lot do. Your cancer therapeutics must be metabolized by the microbiome. And in the UK, 50% of the patients benefit from cancer therapeutics, 50% don't. Guess what? If you actually looked at the microbiome and adjusted that so that it was doing the metabolism of the patients that benefited, you might help more patients. And that's the case in a lot of areas with drugs, is they act first. They see it first, and they deal with it in some way. It can detoxify. It can biotransform food components. So a lot of your relationship to food, drugs, and xenobiotics is dependent upon your microbiome. They can generate agonists for the AH receptors, very potent, again, interaction. They can convert a prodrug to an active drug. They can respond to one drug by changing the metabolism of another drug, which is really insidious, and if you don't know about it, it could be a problem. And again, they can regulate what happens with host metabolism in general. And all of these are things that we can use if we only realize the importance, and we now go to the data that are available on that. So Ellen Silbergeld at Johns Hopkins and I published a paper in Toxicological Sciences a few years ago because Ellen was on the National Research Council that developed the model that we use for environmental health safety, environmental health risk, because it determines when you have an external exposure, what you go through the process of internal dose, biological effective dose, down to altered structure function, and then clinical disease. And you can ask, well, what's the risk? Do we need to regulate this or do something about it? And this was fine in 1987. And again, she was on that committee. But in our paper, we said, no, here's what gets exposed first. You have no idea what's going on down here if you haven't measured it. Because it's all dependent on the microbiome, whether it's in the inhalation, dermal, oral, urogenital. You don't know. So you would think, I mean, it was basically, I was saying, it's great news for toxicologists, you get to do everything over, because we didn't do it this way. On the other hand, we don't know what's safe. And actually, the paper got very little pushback. I mean, it's just a reality. It's the biology of where we are now with humans. So the microbiome can affect environmental chemicals, drugs, and determine, again, the extent to which we're protected against that toxicity. It can determine whether a drug is efficacious or not, or toxic. And the physiological response to foods. So you want to have a better relationship with food, you may want to adjust your diet, but you ought to be adjusting the microbes that are going to be responding to that and want to see those food components as their energy source. So here's an example of one drug. I'm just showing you one. I have talks that are just drugs, drug metabolism. But here's an example of one. Digoxin, the long-standing heart medication, right? Heart disease medication. Physicians know it has a narrow efficacy range. In some people, it's a very, very tight range. Otherwise, it's ineffective or it's toxic. It could kill you. Well, it turns out the whole basis of that range of ineffective, effective, toxic is based on the extent, the population of one bacterium in the gut. This particular species. So obviously, physicians would want to know, if they knew that, you reduce the risk of killing someone or of having it not work. So this can actually rescue drugs that have side effects and problems, existing drugs. But certainly new drugs should not be ignoring the microbiome. We do that at our peril. Okay, so I'm going to talk about the immune system largely, but I can't do a talk and not talk about neurodevelopment and neurochemistry. And part of the reason is prescription drugs and what's being done at the moment. So I have to show you this because there's also a wonderful book by John Kreinen, Timothy Dinan at University College Cork, and Anderson, who wrote this with him called Psychobiotics. It turns out that your gut, and we've known this for a long time, your gut makes more neuroactive peptides than your brain. And a large percentage of those made in the gut are made directly by the microbes. And the rest are regulated epigenetically by the microbes. They're made by gut epithelial cells and the like. So if you want to change your neurochemistry because of major depressive disorder, which is an inflammatory condition largely anyway, or you want to help and benefit neurobehavior, you should go here first because these are existing bacteria that you can change the population and change the neurochemistry and not go first to SSRIs, particularly in children. So this is something where, again, the natural, holistic route is there, and this is actually the most potent. Why wouldn't you do that? Now, whether you can patent it or not, I don't know. This is the way to go. And there's a lot of cases where this is going to be the situation. So I do want to talk about the microbiome and health and disease, and that is that these early seeding events involve what are called pioneering species. These are ones that get established in vaginal delivery, skin-skin contact, and with breast milk. And these pioneering species are really important in converting the baby's immune system from one that has come out of the mother, where some reactions in the immune system were essentially prohibited, because you don't want host-graft reactions. The fetus is genetically foreign to the mom, and you don't want that to be a problem. And one of the ways there are a lot of strategies and components to protecting that relationship, but one of them is that you have the immune system developing kind of in spurts, and not everything develops at the same rate in terms of your response capacity. So the baby has to change so that the baby can actually respond to viruses and things like that. And the baby's out of mom after birth. And the baby coming out of that relationship, if the microbes are not there to interact with the immune system, you are set up for a pro-inflammatory response. And eventually that's going to catch up to you in terms of pathology and disease. That's just the reality of it. And I'll show you the foundation for that in just a minute. So these foundation species are important. They have to be seeded, they have to be fed, which breast milk's involved with that as well. And if you have different populations there, you are going to have different immune maturation. The immune system's not going to mature the same way, and you may have some issues with balances of these cells. Now, I'm going to talk about milk microbiota and human milk in general, but I just want to show that some of those microbes that are in human milk and are available to the infant also influence immune maturation in this post-birth window. So it's important to keep that in mind. The relationship between the microbes, the exterior part of us, right, the gut's open to the environment, the barrier, and the immune cells underneath is very special. And I've actually, in the literature, not that I'm worrying whether it takes hold or not, but I gave it a name, the microimmunosome. And the reason is I want people to focus on the systems biology of this unit. Safety really should be based on this unit, because if you get a leaky gut, if you get gap junction problems, and you have toxins from the bacteria like lipopolysaccharide, LPS, that are leaking through that, you are going to have inflammatory responses that shouldn't be happening in the underlying immune cells. And that eventually will likely, if it keeps going, and is to too great an extent, will translate to organ problems. So what I want to point out is, again, I mentioned that while we were obsessed with the thymus when I was a young professor here at Cornell, but 2/3 of the immune system is in the gut. And that's where, actually, the microbiome, it seems to, the location, it seems to sort of be the master locator. Not that the skin and the lung aren't important, but the gut microbiome is really important. We know more about it, so that's a little unfair in a sense, but it's really important. I just gave a lecture in Albany on risk of Legionnaire's disease and Legionella bacteria, and it turns out that things involving the gut and inflammation from the gut will put you at greater risk. It affects the lung status as well. So you want to protect the lung microbiome, protect the gut microbiome too. It would be easier to do. So 2/3 of the immune cells are here, and they're getting signals from the microbes that are sitting just one cell thick lining away, plus mucus. Mucin being very important, 2 layers. Very important. That's a unit, and we want to protect that. And you can even show that blood-brain barrier changes. If you change this relationship here, you change blood-brain barrier in development. So this translates to lung, to blood-brain, to other barriers as well. So to show you how important and almost sacred the microbiome-immune interaction is, there are 2 really wonderful examples in the literature. One is in 4 species of parasitic wasps, and they've been studied by Rupert Vanderbilt, Bordenstein's lab. And the other are 2 subspecies of the house mouse, the common house mouse, in Europe. And they're divided geographically. So it's really interesting. What happens in these 4 species of parasitic wasps is if they're mated, in the larval, one of the larval end stages, the immune system doesn't like the microbiome that is in the hybrid. And it mounts a massive inflammatory response and kills the larva. Self-killing. Right on the spot. And if you treat with antibiotics and obliterate the microbiome in those larvae, they go on and develop. You don't have separate species anymore. You don't have a healthy wasp, but you don't have separate species. That's a species barrier. The microbiome-immune interaction can define a species or not. It's pretty sacred. We thought that was only due to chromosomal genes in the past. So, in the house mouse, there are 2 subspecies. What happens when they get hybridized? Because they're generally not in the same geographical area. They've been kind of separated by geography. And what happens is they go on and grow, but the microbiome is incredibly reduced in diversity. So both hybrids have a really rich microbiome. I mean, both parents, subspecies, you make them microbiome in the hybrid stinks. It's not diverse at all. And the immune system, it turns out, is not liking stuff. It's killing it off. And eventually the immune system produces massive gastrointestinal inflammation. And those animals don't do well. You know, they may grow a little bit, they may survive for a while, but they are heading for inflammatory bowel equivalent out the kazoo. Okay, and that's, you've got to have a microbiome immune system that grow up together, are blended, suited well. There's pruning that goes on both directions. You know, they're communicating across that barrier, but it tells you how important this is. It literally can define a species or not a species. So that's pretty fundamental. So how does this relate to inflammatory chronic disease, what we call non-communicable diseases and conditions? Well, I just want to point out that these are actually really important in terms of human health. They are estimated by CDC to be producing 3/4 of all the deaths in the world at the moment. And within just over another decade, it's estimated that they'll cost us about half the annual net worth of the world, which we can't afford. Plus, a lot of these conditions produce disabilities, require caregiver status. We're running out of caregivers as a result. They do things that keep us from socializing. If you plan a dinner now, I have my own food accommodations that I require. How many of us do? Even a child's birthday party that our grandparents used to put on, it's hard to do these days. That's us that's changed. That's us that's changed. So, and they accumulate. If you have one NCD, it begets other NCDs as you age, like I do. And polypharmacy along with it, because each condition has prescription drugs and requirements to go with it. That is not a sustainable track. So I have in the human superorganism book, the obesity tree. And I just selected one. I'm going to select one, and it will help us when we go to the flip of the coin, the other side of the coin, and that is colonization resistance, infectious disease. So with obesity, imagine childhood obesity, for example. What is ahead for that cohort if we do nothing different than we have today? 32 comorbid conditions, 12 cancers. If we do nothing different. That's what the literature tells us. Now that's not an individual's trajectory necessarily, it's the group. They're at a higher probability of these diseases and conditions. Well, I'll show you one way this happens. It's not that that's the only way to get to obesity and subsequent problems. But one of the ways it happens is your immune system's responding to the changes in metabolism, the changes in the gut microbiomes. If you're eating a bad diet, you don't have fiber and you have a high-fat diet, and maybe you had a cesarean delivery and formula-fed, all of that combined will cause polarization of one of the immune cell types, macrophages, that are underlying the barrier. And these will take up residence in fat cells. And they actually help contribute to white adipose tissue that is storing fat and is really adverse for us in terms of metabolic syndrome risk. So this M1, in fact, white adipose tissue becomes largely a macrophage organ, an M1 macrophage organ. And this is a highly inflammatory situation producing pro-inflammatory cytokines. If you were to beige this, and you can do this actually in mice, you can flood them with M2 cells, and you can actually make them insulin sensitive instead of insulin resistant. You can actually reverse engineer, change their metabolism by beiging adipose tissue through the macrophages. It's not necessarily the only way to get there, but it is one. It has been done experimentally. So this is a problem not just for the obesity state and metabolic syndrome, but what is now known is that from this adipose tissue, this M1 polarization spreads. So it will spread to muscle, to the pancreas, and to other organs. And so that's one of the ways that you may see these comorbidities, is the spread of immune imbalance and inflammation that will literally spread from the white adipose tissue to the liver, to the muscle, and to eventually that combined with the microbial dysbiosis reach impact in other organs. So one of the reasons I picked obesity and talking about bacterial toxins getting through macrophage polarization is that it relates to the flip side, and that's colonization resistance. So this shows resistance to Clostridium difficile, hospital-borne opportunistic infection, life-threatening, requires antibiotics if you get this, and so you lose colonization resistance from normal microbiota, and C. diff can develop. And then this is going to require antibiotic treatment. But you have a higher risk then of recurrent C. diff because you've got nothing to repopulate the microbiota that was providing colonization resistance. If you do nothing different, that individual's at greater risk for that, besides the other diseases that may come from a depleted microbiome. So it's not that you shouldn't use antibiotics, you should. This is life-threatening. But we need co-therapies that recognize the importance of the microbiome, both from colonization resistance and protecting against other conditions as well. So this is Cliff. I'm in the vet school. I have to show you an animal. It's almost obligatory. Cliff is trained to detect C. diff. And I want to point out that if you don't know where your microbiome status is, go ask your dog. Your dog's sampling all kinds of orifices and can probably tell you something if you can only communicate that with your dog. So C. diff Cliff can pick up individual and hospital epidemics quicker than most instrumentation. Okay, so there's some recent wonderful reviews from the Paymer group. This is one that came out within the last 2 weeks, actually. That was fortuitous. And what they're showing, this is the mucus layer I mentioned. There are two. There's a very dense one and a less dense one. And regulating this is absolutely critical. So here is how colonization can work. And again, Peg Coleman, who's here, could tell you more about it than I can. But it outcompetes pathogens, for example, for nutrient source. It can stimulate through the PPAR gamma signaling. It can signal increases in that will block nitrogen availability for those pathogens that want to use nitrogen. It can stimulate mucin production. I'll show you one of the bacteria that does that. It can directly produce antimicrobial products like bactericid. They're effective against some pathogens. Through TLR pathways, it'll stimulate Reg3 gamma, which is an antimicrobial lectin against gram-positive bacteria. It produces short-chain fatty acids like butyrate, and they can directly block some pathogens, and they can change host defense by promoting T regulatory cells in the underlying layer as well. They're wonderful, wonderful regulators, it turns out. And finally, metabolism by the commensal or mutualistic microbes in the gut of bile acids can be an important route to produce secondary bile acids that can block certain pathogens. And one of the most recent I saw, I didn't get to include it on the slide, is that there are signaling molecules that they can produce that will actually block quorum sensing capabilities in some pathogens. And that's extremely advantageous. That's a fairly new finding. So you've got multiple layers of ways that your commensal mutualistic microbes in the gut, in the airways, in the skin, urogenital tract, naturally block pathogens from getting a foothold. And we need to be using that as the first line defense and using that as a strategy rather than than just assuming everything's fine and we're going to prescribe antibiotics or some other medications. This should be the first thing that we do and then use our drugs as we need them after that. This is just to show you some of those things I talked about. Here's some commensals that can block this particular pathogen in its pathogenic form. Here's the Reg3 gamma that can be produced as well out of the epithelial layer. Blocking these. Mucus production is really important. You degrade that, you're at risk for this one cell thick layer. And then over here we've got the bile acid conversion by this particular commensal against C. diff. You've got sialic acid succinate as products from commensals that can block. And then finally over here butyrate again directly and through the PPAR gamma, blocking nitrate availability for that particular pathogen. So these are just examples that Paymer's group has shown. And I want to point out that this is not just a theoretical situation. I was actually in a department that doesn't exist anymore, the poultry science department at Cornell. I was hired in there in 1977. And in the 80s, maybe extending to the early 90s, we had a salmonella problem in New York State. And it was horizontally transferred among birds, and it was vertically transferred. It was in the egg, and it was potentially zoonotic. So people stopped eating eggs, which hurt the agricultural sector in New York State and elsewhere. And the solution was using something at the time that was called the Nurmi concept. It came out of Finland, Nurmi. That was a researcher's name. That you could have colonization resistance or competitive exclusion and block pathogens if you had the right bacteria in place. So they started loading up these birds with Lactobacillus acidophilus. And it worked. It solved the problem in the 80s. And it's been used for more than 30 years in terms of a sequence of commercial products. And the poultry industry only has pennies on the pound or dozen eggs profit. So before they buy something, it's got to really be worth it. And this is paid off over decades. It's the same principle in humans, really. But this is where there's money, livelihood on the line in a product where you control the whole environment of that bird. So you can load them up with wonderful commensals and really reduce the risk of salmonella, both for the bird and for humans. Food safety. So how many commensals does it take? How many species of bacteria does it take? And this was a wonderful experiment done in mice, germ-free mice, but with a human pathogen, the Salmonella enterica, a cerebrotoctin murium, human pathogen, and I asked a question using metagenomic and metabolomic guided mixes of commensal bacteria. And they said, what could we construct that would provide the best colonization resistance? What would we need? And you might think, well, there's more than 1,000 species in the gut, so it's going to be a lot. Well, it wasn't. It's 15. 15 constructed to provide the complementary metabolism in the niche that would block salmonella. Now, you've got more pathogens than salmonella, but that is incredibly encouraging that that's all it took. So we can do this. We can use metabolomics to guide us, understanding what these bacteria can do, and optimize colonization resistance. So finally, the breastfeeding. I want to point out that breastfeeding has been very effective in terms of risk reduction for a variety of the NCDs. So here's overweight, obesity, here's diabetes, just very recently asthma, but with childhood asthma being the highest level of protection present. And in addition, it does some nice things like reduce telomere length in later age children, which is, again, one of the measures of longevity that's used, or potential longevity. So it's protective in a lot of different ways. I'm going to skip this for time. So breast milk itself, and I hope we'll be talking about this in more detail, provides microbes. It's some of the founding species for the infant. And breast milk changes in composition depending on the age of the child. It actually, each week, is a little bit different in terms of composition. Colostrum is different from breast milk, and breast milk matures and changes in terms of what mom is providing to the infant. But in addition, there are receptor molecules that are important. There's immune cytokines. There is immunoglobulin, IgA, very important in mucosal tissues. And there is this group of carbohydrates, complex carbohydrates, that we can't digest but are the food for the microbes. So they're extremely important, called the milk oligosaccharides, human milk oligosaccharides. And these help to control and establish and help to grow the infant's microbes so that the immune system and other physiological systems, the brain, those neuropeptides, neurochemicals, mature appropriately. And we know that these microbes get established. So this is a plot showing the establishment, and particularly up to 6 months, they're very effective at colonizing. You may still have benefit of those microbes metabolizing in the gut, even if they don't colonize, but up to 6 months they can measure the effectiveness and the difference in terms of mom's microbes through breast milk colonizing. Akkermansia mucophylla is a mucin regulating bacterium, and that is extremely important in protecting that mucin layer. So that is really what separates those toxins from reaching the epithelial cells. And if you degrade that, it's a route to obesity. So guess what breast milk has? It has this bacterium. It's one of the important ones. So I mentioned these sugars being really important, and they feed microbes that then help to prevent these toxins from activating and producing pro-inflammatory cytokines. So you get more Tregs, less of this Th17-driven Th1-driven pro-inflammatory cytokine situation. So this is, I'm going to end with, really, this was a slide I published in Neo Reviews, and I put in a little extra for you, just for you, Peg. So it's adapted from, but this is my worst case scenario. So as a toxicologist, I have to throw in a little bit of everything, but this would be, well, what could go wrong in your child that could really put it at greatest risk? And so you could have, mom has pre-existing conditions sometimes and medications. And I have lectured to OBGYNs and advocated, you know, you've got an opportunity for 2-generational health benefit if you'll pay attention to the microbiome. Help the mom's microbes, and prepare what's going to be seeded to the baby at the same time. So you have this situation, could be diabetes, obesity, restricted microbiome, and then you have stress. And I've used bereavement. Bereavement's a massive stressor. It will change the microbiome. It's a huge stressor. A lot of things can change the microbiome, but that's one. And you may have an urban environment, living near traffic. Your risk of pediatric asthma is directly related to distance from a major traffic artery, for example, during pregnancy or early infancy. So PAHs, air pollution, urbanization, environmental tobacco smoke, and what they will do is reduce your Tregs. So you have more of that Th17, Th1, less Tregs, elevated risk of allergy, autoimmunity, obesity, cardiovascular disease. And then we have cesarean delivery, and it's a surgery, so antibiotics get administered. Reduce microbial seeding, deplete microbiome. Reduce colonization resistance, increase risk of infant infection. And then we go to the question of breastfeeding, breast milk, both the microbes and the oligosaccharides, hopefully, the immune components, colostrum, and if that's missing, missing microbes, missing growth of founding species, altered metabolite, altered immune development, improper control of inflammation. And then finally, we have some of my favorite toxicants, bisphenol A from plasticizers. Again, it used to be in baby bottles, nipples. Europe's been more aggressive regulating it than the US. Heavy metals, some of those are obesogenic, and certain food additives. And they will just continue to process in terms of risk. So failure to self-complete is really one of the most important issues in terms of risk, maybe the single greatest health risk across a lifetime. And in early life, that's our best chance to do something useful. That's where we can have the greatest bang for the buck, so to speak, in prevention. Not waiting until you're my age. Management of the second genome. It's our majority. 99% of our genes microbial. Let's put the right ones in and use them. We can control that. And food should support the whole human. So we need to be feeding the microbes, feeding the right microbes, and understanding that relationship. Because the microbes can control your brain function, and they can make it hard to change diet. I tell people, if I grew up on fried chicken and Mexican food in San Antonio, Texas in the 50s, and I want to eat kale now, I better work on my microbiome as well as just shoving kale into my GI tract. Because otherwise it's going to be a hard sell on my brain. The immune system and microbiome need to co-mature, and that's the way to avoid immune dysfunction. And these diseases, both infectious diseases and chronic diseases, safety has to be for the whole human. So my colleagues in toxicology and the like need to be measuring risk to the microbes, all the way from pesticides to growing crops to food additives. Some of the major food additives that we have damage the microbiome. We know that now. And finally, benefit risk determinations need to include the microbiome. So I apologize, I've used a couple more minutes here, but I appreciate your patience and hope we can have discussions later on. Thank you so much. Thank you.
Just a little bit, as Peg said, I'm a social scientist. I'm a professor of communication. I've been studying risk communication for quite some time. Actually, I started going to SRA as a graduate student. When I was at Cornell, I did my PhD at Cornell, just down the road in the ag school, and I started going and was interested in this issue of risk communication and understanding risk perceptions and how people respond to information. And so when I think about Rodney's presentation, I look at it through the filters that I have, that I bring to the question of understanding how do the words, how do the messages, how might they influence people's responses. And so I'll give a few observations and then bring it to a case study and then wrap and turn it over to the group. So I spend a lot of time in my own research and teaching thinking about how people respond to information, and particularly information about health or environmental or scientific risks. And over the years, the contexts have ranged widely. I'm sort of drawn to things from, one year it's nanotechnology and ag biotech, another year it's ocean health and marine disease biology. Zoonotic diseases are one that I've got a real affinity for from a systems perspective. But in some ways I am context agnostic, although I do perhaps have a bias in choosing ones where I think I might be able to help make the world a better place. That said, in thinking about how people respond to information, sort of the sort of principles, you look at the audience. Who are the receivers of that information? Who is the source of that information? What's the actual message or the content and words, the information that you're providing? And then what's the channel? So in the case of say, just deconstructing this current presentation, we've got the source, Rodney, a professor, a well-esteemed scientist in this area, is providing through a channel of an interpersonal face-to-face setting of an audience that I'm presuming is largely folks that understand a lot of the terminology that you're providing at a higher level of toxicology or science or profession. And then this question of what's the message and the content and the data. It's very much a scientific presentation. You developed your presentation for an audience that would be of an educated level, that would understand this level of science and data that you were providing. So much so we can also, or as such, we can deconstruct other ways when we think about how we communicate to people about certain risks. I'm often thinking about it in relation to the public audience or the lay audience, so not necessarily who's sitting in the room today, although for any given topic we may be an expert in one domain and we're very much a lay audience in the other domain. And so some of the same things like how our values and risk perceptions, our knowledge, comes into play in these different realms. And so, in other words, as scientists, we're not immune from the role of how values and emotions influence our response to information. We may sort of use it or be more aware of it than others, but we are just as much, we have just as much influence. So knowledge is important, but values can influence how we interact with that information. And we also need to think about the social structures around people, because that influences how they respond to information. So their trusted sources, their doctors, their family members. And then we need to think about the institutional role in terms of the larger cultures at play, the laws, the regulations. All of these are going to influence how people are going to respond to information. So let's get to the meat of the matter and talk about some questions that pertain directly to today's presentation. And so from a perspective of a risk communicator or somebody, you could pose the question, and this is not meant to be offensive, but why should people care about the microbiome? Why should people care about microbiota? And I have to say, I don't know if you guys ever check out Google Trends, but it's a handy little thing that you can search, and I plugged in just how many people over the last 5 years in the US had searched for the term microbiome or microbiota, and actually it's increasing. So maybe that makes you feel good. Of the two, which one do you think is the most frequently searched, microbiome or microbiota?
Microbiome.
Microbiota.
Is it?
It is. It is. And if you were to guess, this is also just kind of interesting in relation to, if you were to guess where people are most likely looking for the term microbiome, if you were to guess states.
California.
Massachusetts.
Ah.
And Vermont. Now you can take meaning from this perhaps depending upon what you understand in terms of whether it's health food or whether it's breastfeeding or milk or other types of things, but it's just sort of fascinating, I think, to try to understand who cares about these issues, who's looking up these issues, and what does it matter? Because if we raise awareness about this and you get people's attention, then what is the ask of this. And I think you've provided a lot here. In terms of testing for microbiome safety, in terms of looking at antibiotic use, and in terms of food, in terms of birth and delivery, in terms of breastfeeding. These are just some of the things that I could say you could translate into asks. When you get people's attention, then you need to think about what's the goal of the communication message that you want to send. And in terms of messaging, one of the things that I think that you've got on your side in terms of raising awareness, because what we would understand from risk perception and also communication is that it's really hard to get people to care about things that they can't see. I mean, just think of radon. If you've ever known or heard anything about radon, that odorless, colorless gas, which causes a lot of cancer, it's hard to get people really upset about it. And so eventually it just sort of becomes a part of the law that when you buy or sell a house, you have to have a radon test as part of it, because people themselves, they're not really thinking about radon. So the question is, who's really thinking about a microbiome, right? But you've got that health angle. And that's what we understand from research, is that when it comes to issues related to public health and well-being and disease, you can generate more attention. And so indeed that's helpful when we're thinking about how are you going to get attention to this. But I think that the other point that we need to consider is how do you make sure that people keep paying attention and don't turn away? And in your presentation, I thought that at one point, I felt a little bit more of the rapid heart rate and a little bit more of the oh no, we are sunk. I mean, we have lost so many biome already, and look at all the things that we're doing, and look at all the ways that we are dying prematurely because of this. And it has to, from just stepping back and putting on my public hat, it's sort of like microbiome, superbugs, climate change, terrorism. I just don't want to think about this, right? And so that is the challenge. That's an inherent challenge, and I'm not picking on you in particular, but I think that as risk communicators, when we think about how we want people to care about our risk, we have to understand that they're also getting bombarded with a lot that's going on, and people have this sort of it can have this what's called a narcotizing function, if you will. That's that social science term, sort of bastardization of a scientific term, to say that people become numb to it all. And so thinking about how do we respond to that? And that's when you swooped in your presentation and you said, but here are things that we can do. And I thought that was a very helpful way to provide that sense of efficacy, because what we know also from risk communication is that if you just provide messages that scare people, and you don't provide information of what we would call efficacy information, then people are going to turn off. They're going to not want to hear the message. And so you can't just scare people, because they will turn away from you. You need to also provide them something that they can do. So again, what's the ask? What are the ways that you can provide them with tools to respond? And the last thing I'll say is commenting on the case of the breastfeeding and the pregnant women. And I want to sort of walk through this as a scenario, because I think that it provides us an opportunity of a strategy and something to do. But I think that there's also cautionary things that I just would throw out there for us to consider in relation to this larger message environment. So one of the things as a risk communicator that I'm very interested in are the ethics of risk communication, and the idea to adopt something from medicine, the adage, do no harm. And there's a great quote, I think it was an article, I think it was a book, Granger Morgan, Bostrom, I'm forgetting the authors right now, but they said, we should no sooner release an untested risk message out on the public than we should release an untested drug. But we release so many risk communication messages out there that aren't tested, and so we don't know what the potential unintended consequences can be. And so just thinking through the story of the scenario of the pregnant woman and all of the many sort of ways that pregnant woman can doom her child to an early death or to obesity or to all of these problems, sometimes maybe even before she knows she's pregnant. I think that, we have to think about how do we frame this message so that there's a way that we can communicate this and that it doesn't instill more blame on an individual for things over which they may not have a certain level of control. And I think that it's not to say that that's not an important and a significant target to look at, but in thinking about the exposure of the mother to these different toxins or sources, things early on, are we providing enough of the social support and the structures and the institutional policies to make sure that from an early time people have access to the best type of healthcare and medicine? Are we understanding that how are we look at this in the context of unplanned pregnancies versus planned pregnancies? Are we making sure that we're also not creating a subclass of the unplanned pregnancies that somehow didn't get the message and so therefore are further doomed to poor health or life or well-being? How, thinking through the issue of giving birth, understanding how do we, again, compensate that, yes, C-sections have been on the rise and there's lots of voluntary C-sections that shouldn't be happening. But for some, it does happen, and so we don't make the mother feel guilty that somehow, again, she's doomed her child. What are the ways of messaging around that? And then in relation to breastfeeding, and and I think that there's been a lot of attention toward how do we create structures that support breastfeeding. I was in Minneapolis airport a couple of days ago, and I saw my first breastfeeding pod. Have you guys seen those? That you could go in, and that's and it had some of the information on the side about the importance of breastfeeding, and that is definitely new to me. For me, when I was breastfeeding my child, it was going in, and traveling. It was the bathroom stall at Detroit, airport, which is not a pleasant environment. And so again, but thinking about the social structures and the institutional structures and the support for this, and trying to understand and empathize when those aren't in play, how do we ensure that people don't feel blame for something over which they may have very little agency and efficacy? And so that's just kind of putting it out there of things that I, as a social scientist and risk communication person, think about. That's not to say that you haven't thought about those, very much so, but just sort of putting it out there that when we bring together these interdisciplinary teams, and that's another thing that I love about SRA, is that, where would we normally just be interacting at? I mean, we could run into each other on the quad, but we might not have been brought together if it weren't for SRA, which brings together these interdisciplinary groups to look at these wicked, tricky system-level problems. And so when you have groups together, then you can ask questions, and then you sort of come to a more fulsome solution or way forward. And so that's just another plug for the science and the profession of risk analysis. Those were just some observations, not meant to be critical, but meant to just be some of the things that I thought when I put on my hat as a social scientist. But truly, I just want to say that this was very informative. I learned a lot, and I really appreciate you sharing your presentation with us today.
And thank you, Michael. I didn't acknowledge support from my colleague Michael Roman and Al Pagan, who's running the Facebook live stream for us. Thank you very much. So I wanted to introduce you to this joint project that Upstate New York SRA has been working on with other partners in Australia, New Zealand, New England, and this year in the UK. And we focused on the milk microbiota, or microbiota of milks, and the question that we're asking is, what do we know about the benefits and risks of pasteurization of milks from humans and cows? So if you saw this event on Facebook, you saw this beautiful photograph of the woman nursing her baby in an outdoor environment. This beautiful print of a cow is by an Ithaca area artist, and this is part of a campaign. We're calling it the Whole Truth, Whole Milk campaign, and we're going to start some crowdfunding next month to be able to advance some of the work of this project. So I have postcards at the display table if you're interested in more information about that project. But I really want to move us along to the question period, so I'm not going to speak about the campaign more. But the question I wanted to pose today for our audience is, what do we know about benefits and risks of pasteurization of breast milk? And I decided to offer two perceptions in honor of our risk communication person in the audience and others who might be viewing. But one perception is the potential presence of pathogens in breast milk is certainly a risk for an infant, and perhaps a fatal risk. And the reason I offer this perception is that it's fueling a management decision by human milk banks to pasteurize donor milk. So if you're a mother of a NICU infant and you cannot breastfeed, what's your choice? Pasteurized donor milk. And that's because most of our regulators seem to focus on this perception that just the presence of pathogens is going to kill our babies. Okay, that's one perception. Another one is something that Rodney might be more attuned to, that infants need large doses of microbes, including some small doses of potential pathogens, to develop healthy gut and immune systems. So that particular perception would argue that pasteurized breast milk is a travesty to not give infants the benefits of the full superfood that breast milk really is. So I want to build on what Catherine said about SRA being an interdisciplinary, systems-oriented society. SRA risk practitioners would try to consider the complexity of human superorganisms and also dose-response relationships. So you notice that the second perception had the words doses. And you might not have thought about dose-response relationships for bacteria, but you probably assume that as you increase the dose of a chemical toxicant, you increase the likelihood of the severity of an adverse response. Well, the same is true for microbes, that as you increase the dose of a microbial pathogen, you also increase the likelihood and severity of illness. And what is it that I'm trying to communicate with DRSG on this slide? Anybody know? DRSG is the acronym for SRA's Dose Response Specialty Group. And I have a list of the 16 specialty groups out on the display table so you can take a look. But dose response, I think, is widely overlooked in a lot of our policies for regulating food and health, that we just have not imagined that we need such large doses of microbes would actually be healthful. That hasn't been part of our psyche. So I'm going to just skip through a few slides and tell you that the full bibliographies, as part of this project that's been going on for 2 years now, we have a cumulative bibliography of over 140 papers related to the microbiota of milks, human and bovine. So these are the clinical studies that I found that demonstrated the loss of benefits to NICU infants when they were fed pasteurized milk. So there are a lot of clinical studies in all different parts of the world, and they're all pointing to the same issues that align with Rodney's presentation. Next, there's evidence for microbial presence in breast milk from very few studies that I've found. But here's one study, and it shows a number of potential pathogens that were detected in breast milk. But here are the conclusions about that. That the results of milk cultures don't predict subsequent cultures. So if you're a mom and your breast milk one day contains a potential pathogen, the next day it's unlikely to, or it might contain different pathogens. So that was a surprise to me, actually. Then that random milk cultures, even if obtained at any time during hospitalization, are not predictive of infection in premature infants. So just because a pathogen is present doesn't mean it's going to cause illness. Again, back to that dose-response relationship. Then third, the sporadic nature of the appearance of certain isolates, however, suggests common exposure of both mother and infant. So as Rodney mentioned, the hospital environment seeds the microbiome. That was observed in this particular study. And then here's a clincher. Routine milk cultures do not provide sufficient data to be useful in clinical management. That's pretty, that's a game changer. Why are we using, well, actually, in most of the milk banks around the world, they're not even at that point because they're not testing. They're just pasteurizing everything. But that's the issue that kind of brought us together, looking at human and bovine milks. And so this is the last slide I'm going to talk about, and I didn't leave much time for questions in the main session, but these are the items that we have on the table outside for display. And if any of those listening on Facebook or Zoom would like to email me, I can send these materials. Well, I'm not going to send you the journal or the book because I'm not partnered with this book that has Rodney Dietert's signature in it. But most of the other items that Upstate New York SRA has prepared during this project, I can send you that as email attachments, or you can find it at Upstate New York website. So now I'd like to open discussion and let you, first in the live audience, ask any questions of Rodney or the other speakers. Who has a question? Mary. I'll repeat the question.
Oh, okay, thank you. Thank you so much. That was a great presentation.
Great presentation, Rodney.
But I was fascinated by the slide that you showed of the intestines, and I'm not sure I'm going to get this right, but that the T lymphocytes were reacting against some of the bacteria, killing them off, and then the intestines underwent an inflammatory response. And so you were talking in respect to the infant, but I was wondering how that would relate to adult development of like celiac disease or any that type of thing, Crohn's disease. So maybe could you talk to that? Thank you.
Repeat the question too. Can you paraphrase that for me? Thank you.
So the question, first of all, I got the mic back, so I want to thank Catherine for your insights. Thank you so much for that, the analysis and really important perspectives on public communication, which in the end, that's what has to happen for something to be eventually beneficial and implemented. And thank you. The question was, the immune response in sampling bacteria across the gut lining, and you mentioned T cells specifically. So they're really dendritic cells primarily, and other cells that will sample through and be sampling some of the environment, but they are also interacting with underlying cells, and there's mixes of them in places like the cecal tonsil and as well. So eventually it does affect T cell reactivity and T cell populations. And one of the difference, so they can get involved with the reaction. They may not be doing the sampling as the first line. That's usually more of a macrophage dendritic cell type of event. But they will change, and one of the things that's important is the signaling to these areas. So for example, not for gut, but in skin, there are studies where if a microbe has not been seeded appropriately in early development, that the Tregs simply don't get recruited to underneath the skin. And then you have an inadequate level. And I think this is in mice, I should say, animal model. You have an inadequate level of those, so you're more likely to have an inflammatory problem later, risk of psoriasis or something equivalent, atopic dermatitis. And in the gut, again, in mice and rats, there's a study where there's a narrow window 1 week after birth of a mouse where a bacterial, the Fragilus, has to be in place and make a particular molecule that dampens a cell burst, cell proliferation, that would contribute to colitis later. So naturally that animal, if not affected by B. fragilis metabolism, if it's not, there will be a proliferative burst in the immune cells that later in life, when that mouse gets challenged with an environmental factor, will likely induce colitis. So the microbiome is not just causing things to always come in and to proliferate. Sometimes it has to downregulate or dampen something that could happen otherwise, so that you don't have inflammation, inappropriate inflammation, later in life. And I should explain that inflammation is an important part of host defense, but we want it at the right time, in the right place, in the right form, to do the job and then to resolve, and not to damage, have bystander tissue damage. Sorry, that was a long answer.
Oh, no, that was wonderful. Thank you.
Other questions?
For women that have had cesarean section, so you didn't have the vaginal delivery, have they done any studies looking at taking swabs from the mother or something and giving it to the baby to try and alleviate that issue?
Exactly, and that's actually, I do include that in other talks, but not in a scope that I tried to cover today. It's a lot of territory. Absolutely, and Gloria Domingo-Bellas, who was at NYU, I think she may or may not have relocated to New Jersey, but she was at NYU when she did the work, has done vaginal swabbing, and it is pretty good. It does work. It's not quite 100%, but it is really good in terms of seeing what would be a more normal seeding event, one that you would expect from vaginal delivery. It's pretty close. So yes, and that's just a matter of planning ahead and be willing to do it. There is a risk because of strep. So there is always that risk-benefit issue. That's why this is such a critical forum for those kind of questions to be raised and to address. But as was pointed out by Catherine, I didn't provide some of the solutions with rebiosis. I usually have a rebiosis section, and I myself have personally benefited past the age of 60 of discovering some dietary issues and aggravated inflammation, and being able to adjust diet and probiotics in combination to my better health. So it can be done at any age, just that, well, if you can do it early and avoid the medications and living with these diseases, which is not fun, all the better. So yes, vaginal swabbing, planning so that you can do your best for mom, for baby, to seed and feed the microbiome. Yeah, absolutely. And so that's what we need to be looking for, is in every case. Not everybody, and it's not a matter of blame, it's really the science we had at the time. And we don't want people to feel like anything was their fault, because if we failed as toxicologists and not screening for the right organism, the superorganism. So it was the best science at the time, but now that we know, we shouldn't wait, we should do some things differently. Starting with physicians, patients, interactions, health practitioners, and patients, and people asking the right questions. I know that in my personal case, I will say he's now retired, but my physician was at a more advanced age than I am, and when he saw what happened to my health change, both weight and not requiring drugs that I used to require because of probiotic use, he wanted to read the papers, he wanted to read the microbiome papers, and that's what should be happening, is that education. But it takes conveying it in a way that avoids the jargon, the immune jargon and otherwise. It has to be so it's understandable. Yes, yes.
As a layman in terms of biology and medicine, I had a reasonable idea that the interaction of the human body and the biome was probably pretty damn complex. I had no idea, of course, how complex. You helped expand my mind on that. But that seems to then raise the question to me. I've had some GI problems off and on, and I thought, well, they talk about these probiotics on TV. You should take some probiotics. I took probiotics, didn't seem to do a darn thing. So, and clearly from the indications you gave, that there's a whole lot of stuff that might or might not go on with the probiotics and the interaction of everything else in the body at the time you take them. So I guess you've had good luck with them. I'm wondering if...
Well, my wife and I both, but we used a 3-tier strategy that I probably described it in the book. It's the Wild Wild West. You can waste a lot of money and time on probiotics. On the other hand, there are some very effective ones. But it needs to work for you, or it's no good. So, I mean, the 3-tiered system at the moment, and I should say, you saw that I'm consulting with a probiotics company, actually. They do have products. So I have that disclosure to make. But the 3-tiered system is there research on it? And with bacterial species, strain matters. You want the genes that worked in the experiments and the clinical trials. So Lactobacillus acidophilus is one isolate and strain, is not the same as another strain as another strain. They're different genes with different metabolic capabilities. So that's the first thing, is you want the one that the research was done on, and you want research having been done on it, and you want to know kind of what it might do, what it might change in the environment. And then finally, unfortunately, at the moment, I go to something that I relate to TripAdvisor, and that is, has anybody ever benefited from that? They can sell it, but did anybody actually see any benefit? So if I see thousands of people that did, then that gives me more hope. I might want to risk my money and time on something. So I can tell you that we vetted a bunch. One of the best ones in the literature did not help me nor my wife. It's really good for a lot of people. It wasn't for us. But there are some others that really do help. And the literature supports that. We understand how they work. And so it really comes down to, in the end, a lot of it is, I think, metabolism. And you want to put even microbes passing through your gut. If you're doing it on a regular basis, they might not even have to colonize to be digesting and providing a change in metabolism. So if they colonize, all the better. But you may even see some benefit from flow-through type of system, but they have to get through the stomach. Some of the manufacturers have thought about that, some have. It's an issue they haven't really engaged effectively. So that's important. If they can't get to your gut, get through the stomach, it doesn't matter how wonderful it might be. Did I miss a question down here? Did somebody? I'm sorry. Yes, thank you.
Yeah, so you mentioned that stress in a mother can also cause change in the microbiota. So what happens when we're stressed that would change our microbiota?
So they have been called, depending on the science discipline, the microbiome has been termed, the second brain along with the gut because of the neurochemicals. The endocrinologists have claimed it. They said it's an endocrine organ. So because it makes a lot of hormones and interacts with a lot of hormones, it regulates hormone production. So that's certainly one of the routes that's involved, was changing the hormone milieu. But I can give you an example. It's an experimental one. Rats and mice, they like to run on wheels. That's just part of their activity. If they're in a cage and they're not out in the wild, they love to run. They're happy, they're healthy. You make them run when they don't want to. You stress them. That's a little, kind of a form of torture. You're making the animal run longer, or when it doesn't want to, and you're making it run. That will adversely affect the microbiome. That's all you got to do. So yes, stress, well, now is it a long-term effect? Those are other questions. Is it 24 hours? There's a circadian rhythm. The microbes, you can probably take probiotics and minimize jet lag because the microbes are really tied in bidirectionally with circadian rhythm change. But over-exercise stress, heat stress, in this case exercise stress, they will degrade the microbiome. And if it persists, it can become pretty significant. That's why I use bereavement as the example. That's profound. I mean, we know, I think from social studies, that the surviving, if it's a husband and wife situation, or a married couple, a significant other, the survivor is at risk, particularly the next 6 months. I mean, it's a health jeopardy situation. The microbiome is part and parcel with that.
I've heard that the microbiome may influence blood lipid levels, such as cholesterol.
Mm-hmm.
Is that true? And if it is true, by what mechanism would that work?
Well, because the mix of microbiomes will be essentially digesting your food before the rest of you, digest your food. So what you have in place can either exacerbate high-fat diets and what happens with cholesterol in various ways, or change that up and minimize it. So again, it's the particular mix as food encounters it. You actually could use the same diet differently depending on your microbial mix. And indeed, again, it neurochemically signals the brain. So if I have pizza-grown microbes in my gut and I try to go cold turkey on pizza or pasta, you really don't feel well. I mean, and you don't feel well because the microbes know what they want and they know how to chemically stress your brain and your neurological system. And they do. I mean, those experiments have been done. Chocolate, I tell people, do you have a chocolate craving? Do you want a chocolate craving? Because you probably can arrange that with the microbes. So the rebiosis, I mean, you can change your microbiome profile. But you also can waste money trying to do it. And so unfortunately, the science needs to progress. It is. A lot of people are working on it, a lot of money going into it. And it's going to get better. So I think the opportunities will be better.
Yeah, I just wanted to make a comment on that. That's like the most exciting and most frustrating part of microbiome science. And we know there's a lot of potential there, but we yet don't have the answers. So we all want to get to that place, but there are a couple years of research in between for us to find out what is the best probiotic for me or what diet should I be using.
Right. In the meantime, if we could do things just like make drug safety better and make drugs more effective for more people, that would be an incredible thing. And in some cases, again, we do know the microbes that need to be there to metabolize a drug. I mean, pharmaceutical companies actually can find that out pretty quickly. And so you may not know it, but NSAIDs alter the microbiome, and then you take a proton pump inhibitor to avoid gastric ulcers, and they damage a different part of the microbiome, pretty significantly actually. And food additives like polysorbate 80, carboxymethylcellulose that make your food smooth are really devastating. And they just were never screened. We thought they were safe. Not so much. So there's a lot of, and I don't mean that to scare you, I mean to say we can expect better health because we're going to be able to better buffer and to change things that are in our environment, in our foods, that really, and now we know we need alternatives.
One more question?
Yes.
I just want to know, is there anything I should be concerned about when I eat ice cream?
Well, I think I'll defer. First of all, ice cream can be a wonderful food. I think I had a scoop within the last week. I normally don't, but my wife and I were celebrating something, so maybe making my slides are getting far enough along. But it's not I don't think it would be in the pure food pyramid as the healthiest food around. But on the other hand, things like again the emulsifiers would be the and sugar. Yeah, you probably heard sugar is not too great for our bodies, and it's not too great for the microbiome as well. That's absolutely true. The complex sugars in human breast milk, yes, but sucrose. Well, Catherine, any comments? Again, thank you so much for just so much insight. Well, back to Peggy.
Well, thank you. Can we give a round of applause to our speakers?